So we're finishing up our 5-day series on trying to figure out how neurons communicate, and that's today's last one, which deals with synapses. In particular, we're going to look at how do you continue or are you stopped at the synapse. Refresher, we've been doing this how many times? We have the sodium-potassium pump, move sodium ions out, potassium ions in. Every positive has a negative. So when potassium ions leave the cell through the leak channel, they leave behind negative partners. Potassium stops leaking because it's attracted to the negative inside. We call that the resting membrane potential. If we were to make use of that membrane potential, we call it an action potential. Action potentials we typically look at in terms of reflexes. The job of a reflex is to keep you alive. So that action potential needs to move around. The way it does so is something lets sodium in through a sodium voltage gate. We call that depolarization. That will close those gates, open up potassium voltage gates. Potassium leaves. We call that repolarization. A depolarization triggers another depolarization. The way that we speed this process up is we make it so the action potential jumps. And the way we do that is by adding myelin along the length of a neuron. And you get that jumping sensation. We call that saltatory conduction. Eventually, this is going to reach the end of the neuron, and that ending we call the, well, you can see it says postsynaptic dendrites, or you could call it the synaptic terminal or a terminus, or there's words for it. The point is that at the very end, we have what we call a presynaptic membrane, so the membrane of the neuron, and there's going to be a gap. The name for that gap is called a synapse, and then there's going to be a membrane on the other side, or the postsynaptic membrane. What we have to do is get our information to travel to the presynaptic membrane, jump the synapse, and then continue down the postsynaptic membrane. There's several ways that we can do this, and it turns out that we can use chemicals to do this, or in some cases we can actually use actual junctions that allow for the movement of ions directly from one cell to the next. When we look at the end of the neuron, what we're going to notice is it's not necessarily like everything else, meaning depolarization, sodium comes in, repolarization, potassium leaves. At the end of the neuron, what we'll notice are there's little bubbles called synaptic vesicles, and inside of them they contain chemicals called neurotransmitters. At that synaptic terminal, instead of letting sodium into the cell during a depolarization, instead what we're going to notice is calcium ions are going to flood in. And it turns out whenever calcium ions enter into cells, things tend to move. We get reactions to occur. So the calcium ions are going to come inside of the cell. It's going to cause those synaptic vesicles, those bubbles with the neurotransmitters in them, to travel to the cell membrane. They will fuse and release their contents into the synapse. When they bind, they, being the neurotransmitters, bind to the postsynaptic membrane, we're going to get one of two responses. It can either bind to an ionic gate, and it's going to open up and allow sodium ions to move, or potassium ions to move, and we'll deal with that in a moment. Or, which is actually far more common with the neurotransmitters, it's going to bind to a receptor, which is going to then open up a separate ion gate, but it's also going to trigger a whole bunch of other things to go on. And that is a different story. We're going to just focus on what if it messes with an ionic gate. I'm going to come back to this slide. If, when that neurotransmitter binds to the receptor on the postsynaptic membrane, if it opens up a sodium gate, that means sodium is going to rush into the cell. When it rushes into the cell, we're going to increase the likelihood of a membrane or of depolarization. We're going to reach threshold, we'll depolarize. Well, depolarization is when something happens. And when something happens, it's exciting. There's action going on. So if we move sodium ions into the postsynaptic membrane, we get an action potential. And actions are exciting. So we call that an EPSP, an excitatory postsynaptic potential. If, on the other hand, we're trying to stop an action potential, we're going to try and inhibit it, or have an inhibitory postsynaptic potential, IPSP, what we'll do is have potassium ions move. Because the potassium ions aren't going to stop the sodium ions, it's just going to make it harder for a depolarization to occur. So, neurotransmitter crosses the synapse, binds to postsynaptic membrane. If it lets sodium ions move, we call it an EPSP, because we're going to probably get an action potential. If it lets potassium ions move, we call it an IPSP because it's probably going to inhibit an action potential. At some point, we need to stop all of this, and there's one of two methods that we can stop the neurotransmitter from doing its job. One of which is we can just destroy the neurotransmitter. We do that for neurotransmitters that are relatively easy to make. The ones that are harder to make, what we're going to do instead is we're actually going to use a membrane pump to pull it back into the cell so that we can recycle it. Ultimately, this actually is a complicated deal because there's about 40 different neurotransmitters. Some of them are always going to be excitatory, some of them are always going to be inhibitory, and some of them change their minds. It turns out that one neuron will only secrete one neurotransmitter. So, if you have one particular neuron, it will only make one. So if it secretes dopamine, that's the only thing it's going to secrete is dopamine. If it secretes glutamate, all it's going to secrete is glutamate. If it secretes GABA, all it's going to secrete is GABA. The catch is, depending on where it's being secreted, the postsynaptic membrane might have different styles of receptors, some of which are excitatory, some of them are inhibitory. So then we would say that neurotransmitter is excitatory and inhibitory. In other cases, it will always be excitatory, and in other cases, it will always be inhibitory.